Welcome students. In this video lecture we will study fermentation. It's two types, lactic acid fermentation and alcoholic fermentation, and of course the Cori cycle which is present in lactic acid fermentation, and we will see all the aspects of these processes. So let's start. First of all, what is fermentation? तो fermentation क्या है? Anaerobic condition में catabolism. तो any process that is a catabolic process which degrades or just break the molecules, the larger molecules, into simpler one with the production of energy. Okay, this is a catabolic process. In fermentation, very little energy is released because it is in anaerobic condition. How? Whenever there is absence of oxygen, fermentation takes place, and the main part here is the final electron acceptor is an organic compound, as I have already told you about this in my previous videos. So here you can see that a compound which donates electron and the compound which accepts electron are the same. There are no electron acceptor from outside is involved. So there is a cycle going on where the compound releasing or giving out the electrons is the compound which is accepting it, and it is always an organic compound. There is no Krebs cycle and no electron transport system in fermentation. Only the process which is common in all is the glycolysis. That is the first step of fermentation, glycolysis. It also takes place in anaerobic condition. This is the part which it don't have the Krebs cycle and electron transport chain. And the ATP produced here is by the help of substrate level phosphorylation. There is no oxidative phosphorylation process here. So what is the first step of fermentation is glycolysis, where glucose is broke down and we get pyruvate. One molecule of glucose give 2 molecules of pyruvate, and this pyruvate again has two types of fate. It may have two types of fate. One is called the lactic acid fermentation and other is alcoholic fermentation. In lactic acid fermentation, that usually takes place in mussels and lactobacillus, it is converted to lactate. The final product here is the lactate. The organic electron acceptor is the lactate. So glucose to pyruvate to lactate. In another alcoholic fermentation that usually takes place in yeast and bacteria, pyruvate is finally converted to ethanol. That is an alcohol. That's why this is alcoholic fermentation, and it has two steps: pyruvate to acetaldehyde to ethanol. So we will study first the lactic acid fermentation that takes place in mussel cell and lactobacillus, and here the pyruvate is converting itself into the lactate. Here what happens, another term called Cori cycle is present here, which is present in the skeletal muscle. We will study it in detail later on. It is very important part of the lactic acid fermentation. So this is the process how it goes from glucose to pyruvate to lactate. Let's see how it is done. The path is from glucose to pyruvate and from pyruvate to lactate. The first step, the first process is glycolysis. This you all know. And the second one is reduction, where hydrogen is being added with the help of NADH. So NADH give its hydrogen to the pyruvate and it forms the lactate. So the enzyme here is lactate dehydrogenase. One thing important you should notice, that the NADH produced in glycolysis, actually we have 2 ATP and 2 NADH. So there are also 2 NADH, 2 NAD forms 2 NADH, and this was the gain of glycolysis. But what happens in fermentation, when pyruvate is converted to lactate, again you see that this 2 NADH has been utilized and it has formed NAD+. So Now we have only net gain of 2 ATP. There are no 2 NADH. So there is no oxidation and reduction process in lactic acid fermentation. There is no net gain of oxidation and reduction process in lactic acid fermentation, and fermentation gives only 2 ATP net value. This is the Cori cycle which takes place in skeletal muscle and liver. So how the lactic acid fermentation takes place in skeletal muscle? You can see that glucose changes to pyruvate by glycolysis. Again, by fermentation it forms lactate. This lactate goes to the liver, and in liver it forms pyruvate, and again glucose is regenerated with the help of a process called gluconeogenesis. So this whole cycle is known as Cori cycle that takes place in a skeletal muscle and liver. This is the concept of Cori cycle. Now we come to the alcoholic fermentation. Here it takes place in yeast and many bacteria. Pyruvate is ultimately converted to ethanol, where pyruvate first changes to acetaldehyde and then ethanol. The pyruvate forms acetaldehyde by decarboxylation process, and CO2 is released in this process. And the next process is of reduction, where NADH gives its hydrogen to the ethanol and it is oxidized to NAD, and this is the reduction process. We will see this is the path from glucose to pyruvate, pyruvate to acetaldehyde, and acetaldehyde to ethanol. You see the first process is of glycolysis from glucose to pyruvate, and from pyruvate to acetaldehyde it is decarboxylation. You can see that carbon dioxide is being released. From acetaldehyde to ethanol the process is called reduction. Here 2 NADH is giving 2 NAD plus, and it is being oxidized and acetaldehyde is being reduced. So this is the reduction process. The enzyme for decarboxylation is pyruvate decarboxylase, and the final enzyme for reduction is alcohol dehydrogenase. So here also you can see that the NADH 2 ATP is always there, but the NADH which was produced in glycolysis, it has been utilized in this fermentation process, where acetaldehyde is getting converted to ethanol, and as a result we are having no outcome of the oxidation reduction process. So the conclusion, there is no net oxidation reduction in the alcoholic fermentation. Hope you understand. Thank you so much.